Okay, so you've studied DNA replication and have a basic idea of the process. Now what we're going to look at is what happens when there's a mistake in the process. So mutation is where the DNA sequence is altered in some way. Now there are different ways that this can happen. One type of mutation is just a spontaneous mutation. Okay, so this could happen just on its own. So spontaneous is essentially an error in the DNA replication process. It happens very fast, and instead of a T going into a place, a C goes in place. Even though the C doesn't pair with an A, it just happens. And later, proofreading should fix that. But if proofreading doesn't fix it, then the error remains and it stays as a mutation in the DNA. It's an important part of evolution to create genetic diversity. However, when those mutations occur, you can see they can potentially lead to some problems. So that's what we're going to look at, is what are the types of mutations that do occur, and what types of problems or not occur from them. So if this is our original DNA sequence, for example, up here. So there's two strands of DNA. We have TACCAG, ATG, GTC. So we're going to look at that, and we're just going to be focused on one of the strands that's serving as a template for RNA. And so what's going to happen here is, if this sequence of DNA undergoes normal DNA replication, so this is normal replication, essentially there's no error, no mutation here, the end result should be that these two strands get copied, the new DNA is the same, and then when this strand serves as a template to make RNA, so this is now a piece of RNA here in messenger RNA, it's going to code as a codon for a specific amino acid. It codes for the amino acid tyrosine. That's what should happen. Now, if there is an error, let's say what happens here is there's a mistake. Okay, and instead of the CG pair here, you get T and an A. Okay. So now this strand here, this sequence is read, and you get UA, but then instead of a C here, you have a U. Okay. So that is then called a point mutation. So a point mutation with a single nucleotide is an error. Okay. So in this case, it's this particular one. So now it's read as UAU instead of UAC. In this particular case, UAU also codes for the amino acid tyrosine. Okay. So this is what we're going to call a silent mutation. So technically the DNA has been altered, but there's no end result. There's no change in the end result. The protein that's made, that's coded for in that DNA, would end up with the same amino acid sequence, and it would have the exact same function. There would be no alteration. So even though the mutation is there, it doesn't really do anything. It's hidden, or we call that a silent mutation. Now, let's say when the DNA replication occurs, we get a substitution. It's flipped around. Instead of T on this strand and A over here, it's a T on the bottom and A on top. So now that T now codes for an A. So we get AAC instead of what it's supposed to be. It's supposed to be UAC. That's the original no error. And so AAC is a codon that codes for the amino acid asparagine. So this is now called a missense mutation. Missense, we get a different amino acid put in place of the one that's supposed to be there. A lot of different things can happen. Now, typically, we don't get the same protein. The protein doesn't get the same function. It may not work at all. However, amino acids' characteristics come from their R group. So if it happens to have the same type of R group, they're both, say, basic R groups, it may not make much of a difference. Also, they could have a positive effect. It's also possible that this new version of the protein may have slightly increased binding affinity for a particular substrate molecule. Typically, it probably wouldn't, but this is just a totally random thing that could occur with something positive or negative. You can't think of it as mutation being bad. Mutation just is something that happens, and depending on the particular circumstance, it will either help or harm the organism. And the same thing for the function of that protein. We don't really necessarily know what's going to happen in the end, unless we actually further study it. So missense is when there's, again, a point mutation, a single nucleotide is changed. Instead of an A here, it's a T. So we get a different codon, and that gives us now a different amino acid. So we get a different amino acid is coded for. And there's another type of point mutation that can happen, which is where sometimes slight changes in the code could actually could make that strand unreadable in a way. So what's happening here is that the sequence here is supposed to be ATG has been changed to ATC. So C instead of G, it's the point right there. But what that results in is a codon that is UAG. UAG is one of the 3 stop codons. So that means when this is read in messenger RNA, the ribosome will stop reading the messenger RNA and make no more of the protein. So in this particular case, it's unlikely, whereas this, with the missense mutation, it's possible the protein may still function. Here, you're going to have an incomplete protein, which is highly unlikely that it'll have any function whatsoever. Again, it's possible. Maybe the peptide that results from it has some particular function, but it's highly unlikely. So a stop codon that has been created by a point mutation is called, and this particular one is called a nonsense mutation. So we have nonsense mutations, missense mutations, and silent mutations. So these are also silent, means changing the codon but not changing the amino acid. Missense means we change the codon and then we also change the amino acid. Nonsense means essentially no sense, so we're having the codon changed into a stop codon. And those are types of situations where you have a point mutation. Now, something else can happen. This is where we just get essentially a substitution, right, with this particular type. So where a G is in place of a T or an A in place of a C, that sort of thing. There's another type of mutation that can occur, which is where we could potentially see a deletion. Or sometimes an addition, so an extra amino acid could be added in. These are both going to result in things called frameshift mutations. Okay, so a frameshift mutation is going to do this. Okay, here's our sequence. Let's say this is the DNA up here, the coding, the template strand. It's copied into our messenger RNA here. Now the messenger RNA is read through translation by the ribosome, and this is our amino acid sequence. So you don't necessarily have to know, this is just an example, but you know, tryptophan, serine, valine, isoleucine, valine again, serine. So it's what's supposed to code for. This is the normal non-mutated, or what we call wild type sequence. Now we get down here a deletion. So right here, this one is missing. So it should have been a C, but the C wasn't copied over for some reason. And there could be a number of reasons why that would happen, but it's not copied into the messenger RNA. So now what occurs is that the first two, this codon, still the same, tryptophan, serine, but now when the ribosome is reading, it's still going to read 3 nucleotides at a time, but it's going to be shifting over what it reads. So now we get the UGG for tryptophan, the UCU for serine, that's the same. But now we shift over here. So the C is gone, so now we have AAT, right? Which is for the, then transcribed into the RNA as UUA, which that codes for leucine. So a totally different amino acid. But you see the difference between over here with the missense. We get a single amino acid change in a single location. Every amino acid beyond that point would have been exactly the same, and everyone before it would have been the same. It's this one amino acid that's changed. When you get a frame shift, that means every single amino acid, unless there's some that have the same codon that just happens to be changed into, like the silent one, it's going to be different. So in this particular case, now we have leucine instead of valine, serine instead of isoleucine, cysteine instead of valine. So all new amino acids, so all new or different amino acids, because the entire sequence has been shifted over by one nucleotide, so we have a completely new set of codons to read. Right? That results in a completely different protein, which again may have no function whatsoever in the cell, although it possibly could have some function in the cell. It's just it's not the proper protein that the cell has coded for, that should be expressing right now, and essentially the one that it needs isn't being expressed. That we know for sure. What this one does, we don't know. Could be a problem. It could be nothing. Right? So we have point mutations where one single nucleotide is just switched to something else, and then we have frameshift mutations where we either delete a particular nucleotide, and then we shift everything, or we could insert one, and that would do the same. How would have the exact same effect? It would just be adding a new nucleotide in, and then that shifts the whole entire framework. That could happen as well. It's another type of mutation. All right. Now, what causes a mutation? So, why do these things even occur, in the first place? There are a number of potential causes of them. Some of them are simply like I said, spontaneous. So first off, what would cause it? So our causes of mutation. And we'll get a little bit of extra room here. So I'm just going to clear a little space. I'll take away the wild type one here, the one with no error. We'll leave the ones with the mutation up there if you want to still take a look at those. So what causes the mutation? If it's not just random, just kind of the DNA replication process making a mistake and then having it not be repaired by proofreading, we can have some chemical that we call mutagens. Mutagens, okay? So a mutagen can work in a variety of different ways. Some mutagens will mimic a nucleotide. So for example, it's not guanine, but it looks like guanine, and so it gets incorporated into a new DNA, but then when that's read by cellular machinery, it doesn't know what it is because it's not a guanine, and so it randomly will put some nucleotide there or not. So you either get a substitution or skips it, and then you have a deletion because of it. So the mimics can cause this sort of problem. You can get ones that actually bind to nucleotides. And again, cause them to be misread. Some can add chemical groups like methyl groups, say, to a particular nucleotide, and then it gets read as a different nucleotide. So a G gets read as a C because it has something else attached to it that now makes it look like a different nucleotide to a particular enzyme. All right. So there's a variety of different types of mutagens that work in a variety of different types of ways, often very specific ways, doing both chemical modifications to the nucleotides or mimicking or substituting for the nucleotides themselves. Those are the two major kind of categories. There's other ways they can work, and there's all kind of specific details of it, but that's in general what's going to go on. You also can see radiation. So you get non-ionizing, like ultraviolet light. So this is where we're typically going to break things like hydrogen bonds, and then the hydrogen bonds may reform, and then they may reform in inappropriate ways. So you might get dimers, so a thymine dimer may form, where if you're having a sequence of DNA, A, T, T, G, C, C, A, like this. And on the other strand, obviously you would have your then T, A, C, G, G, T, and so on. Now typically what you would have is hydrogen bonds between the two strands. Okay, hydrogen bonds. The ionizing radiation could break the bonds between the two strands. The bonds should spontaneously reform its positive and negative attractions, but what can happen with thymines is that the thymines can actually bind then to each other, thymines that are right next to each other in the same strand, which means that then when this particular strand, I'll use this here, serves as a template later on, so then they would put a T in here to go with the A, but then the enzyme doesn't know what this is. So we don't know what that is right now because it's the two T's joined together. Are they T's? Are they A's? Are they G's? And then you get an error, right, in the DNA replication process, which puts an error into that gene, which will eventually be misinterpreted when it's expressed as RNA. You also can then get ionizing radiation. Ionizing will create free radicals, and also ionizing radiation can break break covalent bonds. So it can actually cause cuts. Instead of between two strands of DNA, it can actually then cut the DNA itself. So there's phosphodiester bonds between each of the individual nucleotides in the strand. So you should be familiar with nucleotide structure, and it could break those bonds. And then those bonds will not spontaneously reform. So there can be, again, repair mechanisms, things like ligases that can put them back together. But if those aren't functioning or if they don't get repaired, then it actually destroys the DNA. It can be destroyed itself. So and that can cause a lot of other types of problems. Know the basic terms. Mostly it's just really terms here. What's a silent mutation? What's a missense mutation? What is a nonsense mutation? These are examples of point mutations. So they're all examples of point mutations. And then you have frameshift mutations as another type of mutation where you actually get a deletion or substitution, and then it shifts all the nucleotides, and the reading sequence is completely different with all new amino acids resulting. What causes it again? Chemicals, radiation are two very common sorts of things. Right? You don't have to know specific details of the names of mutagens. There are some tables in the study guides and PowerPoints to look at to give you an example of specific chemicals and exactly how they work. But I really just like you to know the terminology.